Okay, let's start this second day. Our first speaker will be by Zoom, and it's Eva Hoffmann from the University of Copenhagen, and she will talk about oocyte aneuploïdy. Do you hear us? Yeah.
Thank you so much, and I'm really sorry that I can't be with you. I'm unfortunately not quite well, and I didn't want to pass anything on, but I'd like to thank Elvan and Marilyn for inviting me anyway, and also making this possible that I can still participate remotely. So today I'm going to talk a little bit about the work that we've been doing on oocyte aneuploïdy and also the implications for fertility here from Copenhagen. So in the lab, we're interested in the genomics of human conceptions, and we have appreciated for quite a while that it is quite inefficient. So we know that after fertilization, about 50% of human preimplantation embryos, they don't actually go on to develop further. And we know that in nearly 100% of these, this is associated with genomic instability. We also know that about 1 in 4 pregnancies are lost. The vast majority, or 9 out of 10, are lost during the first trimester as well. And we're interested in trying to understand the mechanisms that drive the high level of genome instability, and also in turn what this means for the kind of genetic diversity we see in the living population. In turn as well, we also try to study how the genetic diversity that we have might also influence the other way around, the regulation of reproductive traits. So the two curves that we focus on is the fertility curve here in red, and also the natural menopause curve as well. These are correlated with aging phenotypes. Some of them are causative as well of certain aging phenotypes and predispositions. And we try to figure out what we can do in trying to understand the biological mechanisms that shape these curves, but also what that means with regards to personalized approaches for improved fertility treatment. We have long appreciated that the fertility curve is driven by aneuploidies, particularly as women go into advanced maternal age. This is this arrow here. And what we've been trying to do in the lab is try to understand the fertility curve across the spectrum. And we do this by exploring one egg at a time. So we and many others have developed ways in which we can take a single cell or a single oocyte. We can then go in and we can lyse these, and we can amplify the entire genome, going from 6 to 8 picograms into the nanogram range. From there, we then, and this whole genome amplification, we then generate sequencing libraries, or we use SNP array data, and then develop noise algorithms, machine learning algorithms that can detect what's a real signal versus what's noise. And in theory, at the moment, we can detect that we're starting to detect single nucleotide variants where a single base has been altered, and the whole way up to polyploidies with multiple chromosome sets. And I think we can all appreciate, obviously, that when one averages over larger things, it's much easier to actually detect. And today I'm going to talk about aneuploidies. So we take the oocyte and we take the polar body and we isolate them with a laser microscope. We amplify, as I showed you, and what we get out are two kind of mirror images if the aneuploidy arose during the meiotic division. So this is the first meiosis after a very long dictate arrest that we're interested in. And this is a human egg that you're seeing. You can see as well that the vast majority of the chromosomes that you're seeing along the x-axis here, they have a 2C content. That means they have a single chromosome. But you can also see that sometimes we end up with gains and also losses. These are reciprocal, and therefore we know that they arose during the first meiosis. When we do this across lifespan of reproductive lifespan of in human, we see a U-curve, and this U-curve is really shaping our fertility curve. So this is in veal here, and you can see here that euploid eggs really seems to kind of form a fecundity curve above it. I've put in chimpanzees here as a control, and I'm happy to take questions about this. Now, one of the implications of this is, of course, that our eggs don't just get worse with advancing female age or female age in general. A 15-year-old and a 38-year-old have the same incidence of aneuploidies, about 50%. So eggs don't just get worse, they actually also get better up to a point at around 25 to 27. What we also do know is that there's a chromosomal bias with age. So when we have very young females or eggs from very young girls, it's the large chromosomes 1 to 6, that preferentially have segregation defects and end up aneuploid in the egg. On the other hand, as we go into the advanced maternal age, at this stage of the curve here, it's the smaller chromosomes, and particularly 21 and 22, but also 15 and 16. And this is telling us that it's a different type of risk with regards to chromosomes that are making mistakes that kind of give the curve its shape, at least to some extent. What I'm not going to talk about, and I know that many of the people in the audience also work on, is obviously mechanism that might change the entire curve, because even at its best, there's only about 80% of euploid eggs. So that means that 1 in 5 eggs, or 20%, are still actually aneuploid. The other feature, besides chromosomes that shape this curve, is also the mistakes the chromosome make. So this is a collaboration with the Origin for Aneuploid Research Consortium that we did, and this is from a review, but the data as well is from the original paper, now a bit from about 5 years ago. What you can see here is when we categorize the female ages from 5 to 15, and then in the middle age, and then in the kind of advanced maternal age, you see different colors. And that means that different chromosome segregation defects. So in the blue here, you can see that as the girls are very young, their eggs basically mainly contain meiosis 1 non-disjunction errors, as shown here. So both chromosomes segregated to the same pole. As women then get older and into the mid-age and also into advancing maternal age, we see that proportion, actually, that incidence going down. And instead, we see errors coming up that are pre-divisions, so sister chromatids that segregate from each other precociously. So either in this way here, and as women go into the advanced maternal age here in yellow, also reverse segregation as well. And all of these, they give different types of vulnerabilities, aneuploidy risk after assuming a normal meiosis 2 at fertilization. So what we have is a two-factor model in which it's the chromosomes that shape the curve, and it's also the kind of mistakes they actually make that differ between them as well. So one of the biggest issues, I think, and biggest discussions, of course, is do these aneuploidies, are they propagated into preimplantation embryos and also pregnancies as well? And this is these are data from Natera that we've analyzed, and you can see here that these are maternal trisomies that we're picking out. So these are only the gains, either from a blastomere or from a trophectoderm biopsies. So these are after fertilization on day 3 and day 5. In the red, you will see day 3, and in the viola blue, you see day 5 biopsies. And you can see here, the first thing is that we also see a slight U-turn when we go from the kind of optimal fecundity into the young age. And we also see this very well-known maternal age effect when we model these data. So it seems that we're certainly propagating many of the errors from eggs. They're coming, and they are also being propagated into preimplantation embryos. What about pregnancies and pregnancy losses? So there's a lot of focus, of course, on prenatal testing programs that we have in many countries across the world. In pregnancy losses, that was one of the first places that we started studying aneuploidies. We've been working with Henriette Svar Nielsen, who's been leading the Copenhagen Pregnancy Loss Project, a study. We've developed a blood test for detecting aneuploidies in pregnancy losses. And what we've done is to go in and correlate and validate pregnancy loss and circulating fetal DNA from the pregnancy loss in the mother's blood with also the actual pregnancy loss material. And as you can see here, about 45% of the losses are due to aneuploidies. That's the one shown here. Some are single in brown, and some also are multiples. That's what we see in eggs as well, particularly as women get into advanced maternal age. Again, we see chromosomes 15, we see 16, we also see 21 and 22. But we also start seeing monosomies at a high incidence. And one reason for that is because at this stage as well, viability of the fetus and development also becomes important. And that to some extent also determines what we will see. Okay, so having established that eggs and oocytes contain a high incidence of aneuploidies, they're propagated into preimplantation embryos and also pregnancy losses. Is this something that we know is important as well for outcomes in fertility treatment? So this is kind of a study from the HFEA or data from them with egg donation. And egg donation really allows us to go in and first of all, as a treatment, use it. But secondly, of course, also to scientifically look at, is it really the egg or could it also be other factors in the woman? And what you're seeing in the upper panel here are couples where they're using the patient's own eggs. This is under 35, and then the age groups get more advanced, up to 45 and 50 here. And by that stage, under 5% of the cycles actually give a live birth per embryo transferred. However, if we now use donor eggs, so these are eggs from women obtained under 18 to 30 to up to 35, you can see here that now that pretty much resets the efficiency of this treatment. I showed you that there's a U-curve, and that also, of course, raises the question about, well, can donor eggs or could donor eggs also maybe be too young? At 18, I should say that elevation in aneuploidy is still very, very slight. But there have been some studies. This is one of them. This is a retrospective study from Nick Macklin's lab in the UK, where he's been looking at the outcomes of oocyte donation pending the donor's age. This is from about 500 altruistic oocyte donors aged 18 to 35. What he sees is that with the donor age of 18 to 22 years, it's associated with a lower incidence both of clinical pregnancies and also live birth after the first embryo transfer, compared to donors who were a bit older in the mid group, basically when fertility and fecundity appears to be at its highest in human females. This is a retrospective study, and obviously that comes with a number of limitations, but it nevertheless indicates certainly that the U-curve, both for the AMAs, for the advanced maternal age, but also for the young, is important. Okay, so the mechanism by which this might work is the one that we have, at least in the lab, is that we work from, is to try and understand what actually happens on a per cell basis. And we think this is cell autonomous because not all eggs go bad at the same time in a given woman. So in all the eggs of advanced maternal age women, we think that there's been a very good recombination and also establishment of cohesion between the long chromosomes. So during the extended dictator rest, these are decades because these are formed during fetal life. So during this decades of rest, they will be absolutely good, they'll be fine, and they'll be, because there's more cohesion and there's also more recombination, tethering them together, holding them together during 30 or 40 years. In contrast, the smaller chromosomes, particularly 21, 22, but perhaps also 15, they show up increased loss of potentially cohesion, and that would give rise to the precocious separation of sister chromatid that we start seeing coming up in the mid-group. And then finally, as women get into really advanced maternal age in their 40s, we start seeing whole arms in univalence, both in spreads, but also this kind of bipolar orientation now and reverse segregation. And that's really been worked out by Melina Schu's lab. So this is the kind of working model we have. And we've been trying to figure out, can we actually study that? And can we also try and figure out what kind of factors might be influencing not just cohesion loss, but even maybe recombination or other factors that might be important for preventing aneuploidy or determining segregation outcomes, not in the fetal stages, but decades later in the mature egg. So we had a collaboration with Rajiv McCoy's lab and a PhD student, Sarah Karikosias, who just graduated. And together with Natera, we now had access to about nearly 140,000 preimplantation embryos, data from it, and also nearly 23,000 pairs. We could separate maternal and paternal haplotypes. So in the unobserved embryo, we could map both haplotype-specific copy numbers. In this case, it's both purples from the mom, And we could also look at meiosis-specific meiotic crossovers that occurred decades earlier, we think, in the embryos, in the fetal gonads. When we do this, importantly, you can see here that we see the maternal signal for 15, 16, 21, and 22. We see a maternal age effect as well. There's no paternal age effect here. The reassuring thing is that we are seeing a Y chromosome signal only from the father, which makes sense, of course, but also an equal contribution to chromosome X. And that's kind of also well known from Klinefelter that the parental contribution there are similar. So we can also map crossovers, and very similar to what we see in human population, paternal crossovers, they seem to be not so, they seem to first of all be about 1.5 to 1.8 fold lower, and that distribution is also more squished. In the maternal, on the other hand, you can see that there are more crossovers per meiosis, and there's also a much wider spread of recombination events as well. When we do this and we analyze euploid versus aneuploid embryos or genomes, you can see as well that we see lower recombination rates or crossover rates when we have aneuploid maternal genomes. And we think simply that this is because the likelihood of distributing them correctly or failing to cross over entirely might make chromosomes more vulnerable to the very, very extended arrest. This is just a control to see whether the hotspot occupancy is similar, and we don't really see any differences between euploid and aneuploid maternal genomes in that case. So recombination between chromosomes in fetal oocytes, I'm showing you here, affects aneuploidy in adult X and also in embryos. And this is really consistent with many smaller studies, or several smaller studies in human X and embryos. Okay, so I just want to finish off with this last bit. Can we then identify genetic factors in the mother that regulate aneuploidy and crossovers? So this is actually what we have in the human population now. So this is a common phenotype. All women go through it. So we decided to look for common variants, and because we have such a large data set now, well, relatively large data set, it meant that we could start using genome-wide association studies. So we're using single nucleotide polymorphism arrays. We impute about 10 million different genetic variants, and then we use additive statistical models to look at the contribution of each of the genotypes, possible genotypes at these positions. When we do this, we found two signals, and one of the signals on chromosome 22 here, we could deconvolve further. And when you look at this, at the three genotypes here, you can see that we start seeing a separation across the entire maternal age of about 3.3%. Now, this, you will say that's a tiny effect, a small effect, and that's exactly what we would expect from a common variant. We can also see that this maps directly to SMC1 beta, which many of you will know, and I will talk about in the next slide. Because we've known for quite a while that SMC1 beta, from really nice mouse model work from both Jesberger, Pat Hunt, that SMC1 beta leads to very early infertility when it's in the female mice when both copies or single copy is lacking. It's a cohesin ring, meiosis specific, that mediates both meiotic recombination as well as also cohesion between sister chromatids. When we look in gene expression in very large data sets that are out there, we can see already here that it correlates with the different genotypes as well. So that the CC genotype now has lower expression than the other two genotypes. And this is also consistent with that this is the genotype here, the CC, that causes the elevation in aneuploidy independently of female ages. Its frequency is quite high. So you can see here in African population, it's at 78%, and in European and other non-African populations, it's about a third who carry one allele. Now, interestingly as well, SMC1 beta also correlates with other fertility traits as well. You can see here in this heat map here that the age of menopause has now blue, so it's been reduced slightly, and the age of menarche has now been older, is now a little bit of a later age. And that means that the reproductive lifespan has been shortened at both ends for the females carrying this allele. I want to just finish with obviously saying that 3.3% and these effects that we are revealing now, there's no way they can explain, of course, the kind of more extreme phenotypes that we see in women in fertility clinics. So one of the extreme phenotypes are women who, for example, have all of their eggs being aneuploid already in their mid-20s. Others are in their mid-40s or even 50, and all of their eggs seem to be absolutely fine. And I would just like to point you to Karen Schindler's really wonderful work, for example, here last year, of trying to identify and sequence by exome sequencing rare variants, but with really large effects that influence aneuploidy incidence in human eggs and embryos. Okay, so I just want to summarize. I've shown you here today that we have an age-dependent aneuploidy, and it follows a U-curve in human, that maternal age affects chromosomes and also the type of segregation error or the other way around. Eggs don't just go bad, they also get better. And this has implications for fertility as well as pregnancy loss, also I should say incidence of congenital disorders as well, as I showed you data of. And finally, I showed you as well how we are starting to look at the genetic contribution to aneuploidies, both by rare variants with large effects such as those from Karen's lab, but also common variants with very small effects that these very large data sets now give us access to. And I think, I know this is about mothers, but I also just want to say that it's also fathers. This is our paper we had out a couple of weeks ago together with Decode and Henriette's group here. And I just want to say as well that the paternal genome also causes pregnancy loss because there are small structural variants and also de novo mutations, many of whom originate, or the vast majority of which originate in the paternal genome, and particularly with paternal age, that also can explain pregnancy loss, but also certain other developmental disorders in children. With that, I just want to acknowledge all of our participants who participate in our studies, our many collaborators, the lab, and also our funders. Thank you very much.
So we have time for a few questions.
Hi Eva, Tommaso Cavazza here. Very nice talk. I was wondering, you showed this data about the fact that with egg donation, it doesn't really change the birth rate, independence of maternal age, but though the numbers were still around 25%. Do you know from experience or from that study, what are the causes of pregnancy loss in this case? If aneuploidy is still playing actually a major role, or if it's implantation failure, or?
Yeah. It's a really great question, Tommaso, and I think there's a lot of effort going into trying to understand what is considered a euploid loss. So one of the limitations currently now with the whole genome amplification, but also with cost of doing these very large-scale screening prior to transfer, is that the resolution is only at best a megabase, and that's really at best. I think it's probably safer to say it's 5 megabases. So that means that any other contribution at that stage won't be detected. And I think that's important. We know that there are other, for those who don't use nib array-based or at least B allele frequency-based approaches, we also know that about, I would say up to 5% might also actually, probably a bit lower for ICSI. But if you look at IVF and ICSI combined, up to maybe 5% would also have be triploid. In pregnancy losses, 10% of the losses are triploid. So I think that there are things that we're not detecting at the moment. How much is aneuploidy? I wouldn't say there's much more aneuploidy per se. There could be some mosaicism, but that's low. I mean, that's quite low incidence now with better algorithms. So I would say there might be other genetic contributions that we're not seeing. But even for pregnancy loss itself, 45% are not explained by fetal genetics. In this study that we did of about 450 pilot losses together with Decode. So I would suggest that there might be other things going on with implantation.
I'm sorry if I missed this in your talk, but what is the frequency of mitotic errors that contribute to aneuploid embryos, and how does that compare to meiotic errors?
Well, I mean, so there are two different ways of thinking of that. There's the total load of mitotic errors, and the total load of mitotic errors is high because about 50% of human pre-implantation embryos, they actually arrest, and 97% of those, they have errors in them, generally speaking, mainly mitotic, often also chaotic karyotypes. So in that way, the burden is quite high. This is up to day 3. With regards to the ones that go through to the blastocyst, and when you then take a clinical trophectoderm biopsy, mosaicism I think is less than 3% in the biopsy taken. Now, that doesn't mean that there aren't other mitotic events that have happened, but generally speaking, it's quite low compared to meiotic errors from women. That will be up to 80%, in a 42-year-old woman. I hope that kind of addresses your answer or answers your question.
If I understood well, in the population, there are outliers where the aneuploïdy rate is super low. And so my question is why these genotypes are counter-selected, because in some ways it's counterintuitive to have this outlier with low aneuploïdy that are at really low level. There must be some kind of counter-selection, I guess.
Yeah, I think it's a really interesting question. I'm hoping Karen Schindler will start sequencing those. Karen started with the ones who are really high for their age, right? I think we take our inspiration to some extent from the aging field, that if you look at certain aging phenotypes, some of them is, for example, it can be dementia, for example, right? And you have some people who live to more than 100, and they're absolutely fine. When you look at the sequencing and you look at the buildup, for example, of different various morphologies associated with it, it just seems to be that they have a lower load of pathogenic type variants, rather than that they have something protective. And I would suggest that would be part of what you call the deselection here, because if you have a distribution across the population, you would always, for common traits at least, and assuming there's a significant genetic contribution, you would expect also to see outliers. Now, to what extent that's driven by both rare monogenics and up to polygenics and combinations thereof, I think that's one of the things that I'm really hoping that we can start looking at now that we have these very large data sets to study.
You made the interesting point that aneuploïdy gets better before it gets worse. So can you say anything about why it gets worse early? Sorry, why it gets better earlier?
Right. We don't know at the moment. We are looking at various different hypotheses that we are testing also with mouse models. In theory, what we observed was that it's the largest chromosome, so we have hypothesized that this has something to do with size dependency to some extent. This could be that there are additional crossovers, it could be condensation, it could be anything to do with chromosome biology, it could even be it could even be overloading of cohesion, for example, as well, that can't be removed properly. So what we're trying to do now is use mouse models of SMC1 beta, but also in general look at mouse models that are very young to see whether we also see a different aneuploïdy profile compared to those that we wait that are of advanced maternal age. So we're trying to pursue it by going in with a chromosome biology kind of focus to try and find out why they act differently. I should perhaps say that I showed extreme ends where we have large chromosomes and small chromosomes, but you also have chromosomes like chromosome 13, which is the one whose trisomy will result in Patau syndrome, where you actually see it follows more of the age curve itself. So 13, at least in cross-sectional studies, seems to be starting out quite bad, and then it seems to get better, and then it gets bad again.
Okay, thank you. Yeah, Eva, very nice talk. This is Melina speaking. Really nice data. It's very interesting that you see this SMC1 beta variant and that there's also difference in prevalence for different ethnicities. And I was just wondering, have you looked more into this? Like, is there also like an earlier onset of like kind of IVF rates dropping more rapidly, higher aneuploidy rates? And I think that's quite important also for maybe adjusting IVF treatments, giving recommendations for egg freezing. And I feel like maybe that's something that the field should look at more, and probably you've got a really nice data set also to look at this. And maybe, I was just wondering if you see that also reflected in maybe aneuploidy rates, IVF success rates.
We are looking where we can stratify. Also, as you know, Melina, access to IVF is highly socioeconomic factors that go into that. But we are looking at what we can actually get out in a meaningful way. And I think it's exactly what you're saying. It's obviously very, very important. In this case, the SMC1 beta, the allele is really, really rare. Well, sorry, it has a really small effect. And I think as we get larger data sets, I mean, this was just 40,000, right? But hopefully, as we can compile more data together, we can get up to much higher power to start also looking at ethnic specific variants, and also, of course, see if we can correlate that with outcomes clinically. I can say one thing, and that is ethnicity does matter with respect to age and natural menopause. So the IVF cycles as well might also well be affected by, for example, response to stimulation as well. Whether aneuploidy is also affected in a similar fashion, as you know, is just much harder to study because the clinical outcomes, in this case PGTA, is just not available at this, data are not available at the same level. But I'm really hoping we'll get to that.
So our next speaker is Karen Schindler from University of Rutgers, and she will talk about oocyte acentriolar spindle assembly.
All right, good morning. I want to thank Marie-Helene and Elvin for putting on such an amazing workshop. Yesterday was great, and I'm looking forward to today, and thank you for the invitation. When I saw what was sort of suggested that I speak about, I decided to unearth a project that we've kind of been slowly working on in the background, and you'll see that it connects really nicely with what Eva just presented, and so some of the background information will look very familiar to you. All right, so my lab at Rutgers University is broadly interested in genome integrity and egg quality and how that persists over the reproductive lifespan. And we have a number of projects that are focused on answering questions in this area. Some of the work that I won't talk about today is focused on the aurora kinases and how they regulate chromosome segregation, how a sirtuin called SIRT7 is regulating genome stability with age. And we're also really getting interested in cytokinesis and something called a meiotic midbody and how that's connected with egg quality. But what I'll talk about today is our more translational work in trying to understand genetic risk leading to aneuploid conception, in particular the impact of genetic variation on meiotic spindles. Okay, so Eva didn't show this curve, but this is a really famous curve from Terry Hashold and Pat Huynh, showing that with increasing maternal age, we have an increase in trisomic pregnancies. So here's maternal age on the x-axis, trisomic pregnancies on the y-axis, and you can see that at that bizarre age of 35, there's this huge increase in the incidence of aneuploidies. But we know from work from Eva, and this will look familiar, is that with individual patients, these aneuploidy curves are not so smooth looking. So this was data that Eva had published a couple years ago, looking at aneuploidy rates in 8-cell embryos and in blastocysts. And again, you see these nice sort of smooth average curves that at the age of 35, the incidence of aneuploidy increases. But what you can appreciate in these sort of faded foci is that patients are kind of all over the place with respect to aneuploidy levels. And so, not one curve fits all is the way that we're thinking about this. We were kind of stumbling on the same information when we started collaborating with a local IVF clinic, RMA of New Jersey. And back in the late 2000s, what RMA of New Jersey was doing at the time was pioneering doing trophectoderm biopsies for pre-implantation genetic testing for aneuploidy. And the reason why they were doing that is that they wanted to counsel patients to transfer a single euploid embryo to reduce the rates of higher order multiple pregnancies that are of high risk. But the guy that was running, or the clinician that was running the RMA of New Jersey, was really into biobanking. So he was banking maternal blood samples for doing genomic studies at some point. And that's where we came into the picture. So Nathan Treff was working at RMA of New Jersey at the time, and I had started at Rutgers, and he came to me with this data, and he said, you know, what do you make of this? Is this something that we could study together? And again, this is a very similar sort of picture of aneuploidy with respect to maternal age. So here's maternal age of patients undergoing IVF cycles, and then the percentage of her blastocysts in a cycle that were aneuploid. When you plot the average data of aneuploidy for this patient cohort, what you see again is at age 35, aneuploidy increases. So what we've highlighted on here in the empty circles are patients who statistically behaved as you would expect for her age. But the patients in red are statistical outliers, so they either did worse than you would predict for their maternal age or better than you would predict for her age. So we were really focused on the patients on the top part of the curve, but I'm happy to also talk about these patients that Eva kind of hinted at. So our hypothesis, since I was in a genetics department, was, well, maybe there's some gene variants that we could identify in these extreme aneuploidy rate individuals that could help us understand and predict their aneuploidy risk. And so what we started doing is doing exome sequencing of these phenotypic extremes. And I collaborate with a computational geneticist in the department to develop an experimental pipeline to test these genetic variants. So we work with Jin Xing, who does all of the in silico work. So he does a lot of stuff on computers that I can't really explain to you, but lots of bioinformatic prioritization. He comes to me, we talk about the lists of genes that he's ranking, and what we did was develop a mouse expression assay using in vitro overexpression to then screen these genetic variants. Those genetic variants that cause elevated rates of aneuploidy in mouse eggs, and the ones that we're really interested in, we then move into the mouse, use CRISPR to knock those mutations in, and then study the in vivo impacts of those mutations. So in this pipeline, this was an older data set that Nathan had sequenced before we got our NIH funding, and we had data that was on an ion torrent platform. So Jin did all of this analysis and came to us with a list of genes, and one that we were particularly interested in hit this Go enrichment term called centriole. And two of the genes we've started working on. One is called SEP120, and that will be the focus of my talk today. And the other that we've started working on is called LRRK1. I was hoping to talk about both, but as I was putting it together, I realized I would never finish on time. So I'm happy to talk a little bit about that if people are interested. And part of this story, as I mentioned, was published in 2020. At the time, we wanted to make mouse models, but we all know what happened in 2020. So we kind of panicked and decided to just publish this first story. But I'll show you what we've been doing since. So this was the list of genes that hit the centriole term, and SEP120 and LRRK1 are right at the top of really high-ranking variants. Most of these variants are not rare. They're fairly common in the population, except for this one particular one in LRRK1. I'll zoom in a little bit more here. What we do is look at protein analysis to see if the amino acid substitutions that these gene variants make, they could be predicted as being damaging as part of our ranking system. And all of the variants that we identified in SEP120 or LRRK1 were predicted to be deleterious or damaging. So we thought that they could be interesting to work on. So to tell you a little bit more about SEP120, this stands for centrosomal protein of 120 kilodaltons. In somatic cells, it localizes to the pericentriolar material of the centrosome, and largely has been described to act as a microtubule nucleator, essential for spindle assembly. It's been implicated in a number of neuronal diseases, one that in particular called Jobert's, and people are also interested in working on it in kidney function. CEP120 is pretty highly conserved throughout evolution, and it has an N-terminal microtubule binding domain. It has a C-terminal domain with a coiled-coiled motif, and then a dimerization domain. And it's known that this protein homodimerizes, and you need that dimer to be able to function to help nucleate the microtubules. In the C-terminus, human and mouse CEP120 are highly conserved in sequence, and this arginine that's mutated to a histidine, this arginine residue is also conserved between human and mouse. So it kind of fit all the parameters that we like to be able to go forward to test in our system. So first, as a sanity check, we wanted to make sure that CEP120 is expressed in mouse oocytes since we're moving from human into mouse. And at the time, we didn't know if CEP120 even existed in mouse oocytes, and so we purchased a commercially available antibody. We took mouse oocytes and fixed them at metaphase 1. And when we detected CEP120, we noticed that it localizes right at the poles of the spindle. So it's expressed, it goes to where we would predict it goes. And this is important to do because what we then do is we clone the human genes and we fuse them to GFP and make sure that they could also localize where the endogenous mouse protein would go. So here is human CEP120, it's fused to GFP and it localizes right to the spindle poles in a mouse oocyte. And this is important because then we do site-directed mutagenesis and then we inject in the human variant that we identified. And I'm going to skip over a lot of data that's in this publication so I can share with you some unpublished data. But ultimately, what we found was that if you overexpress either wild-type CEP120, we do elevate meiotic aneuploidy a little bit, but if we overexpress this variant of CEP120, we elevate aneuploidy up to about 40%. And this is an in situ chromosome spread that's a little bit more affordable to do than the sequencing, and something that I worked on and developed when I was a postdoc with Richard Schultz and Mike Lampson. So again, this was 2020. We went ahead after everything kind of settled down, and we used CRISPR to knock this mutation into the mouse genome. And the first thing that we did was some long-term fertility trials where we mated either wild-type heterozygous or homozygous mutants, females, with wild-type males. And we just let them hang out together for about 6 months, so they have about 6 or 7 litters over a span of time. And what you'll notice is that in the black and the blue curves, these are our wild-type and our, excuse me, our heterozygous mutants. These animals have pretty similar fertility over time. So the heterozygous does not seem to be affected. But the homozygous mutant, although it started off with pretty similar fertility early in reproductive age, it started to trail off over time and started to become subfertile earlier than an average age for a mouse. And it started becoming subfertile around 5 or 6 months of age. So we wanted to know if this was associated with an increase in aneuploidy. So we went back to our in situ chromosome spread assay. And so what my student Marlena did was take reproductively young mice and take a look at aneuploidy levels in their metaphase 2 eggs. Her wild-type mice have no aneuploidy. There's a slight elevation in the heterozygotes. And then the homozygotes, although it was statistically significant, it's still sitting at about 8 to 10%. So this might be why in the fertility trials we don't see a difference in fertility at the young ages. And then when she took older mice, we see that aneuploidy levels start to come up regardless of your genotype. So our wild types and our heterozygotes both have about 20% aneuploidy, and the homozygous mutants go up to about 35%. We wanted to know if age was playing a significant role with these different genotypes. So Marlena did a 2A ANOVA analysis, and actually we were kind of surprised that age was not playing into this increased aneuploidy, and so it's really just sort of like this global increase that we're seeing with age. So the rest of our studies we actually did with the younger mice, in part because age didn't seem to be significant enough for us to make the investment to hold the animals for 5 to 6 months. So what Marlena wanted to do was ask if spindle building in these mutant mice was affected. So she looked at metaphase 1 spindles, and what you can appreciate right away in this image is that these spindles in the homozygous mutants are a lot shorter than our wild type or our heterozygous animals, and that's quantified here. They're statistically significantly shorter, and they have a smaller volume. So Marlena is an atypical PhD student in my lab. She's actually been an embryologist for 30 years. And in the US, they've changed the rulings for lab directors in the IVF clinics. They have to have a PhD. So she's gone back to get her PhD so that she can keep her job that she does very well. So she really wants to make these connections with the clinic. And she wanted to know, in these oocytes, does this mutation alter microtubule nucleation? So the assay that she set up was to take oocytes and mature them to metaphase and meiosis 1, and then she depolymerizes the spindle by giving them a cold treatment. Then she allows them to warm back up, and she assesses their ability to nucleate microtubules. And so this is the assay. So here are some of her controls. She fixes oocytes before the cold treatment to make sure they're at metaphase 1. They look good. After a cold treatment, completely gets rid of the spindle. That's what we want to see. After recovery, her wild type spindles look pretty nice, but in the homozygous mutants, we start to see problems. We again start to see sort of thin, maybe shorter spindles, but we also saw a number of tripolar spindles. And that's something that we don't see when we just mature these oocytes. It really requires them to have to kind of gear up the system again. And they seem to really be deficient in making quite unstable spindles, something that in the literature we see a lot in human embryos. And so this is quantified here, statistically significant reduction in spindle volume and this sort of increase in abnormal spindle building. Okay, so this is really where she starts getting connected to the clinic. So she said to me, she's like, Karen, one of the things that we do in the lab is we freeze eggs and we freeze embryos. I wonder if there could be an issue if a patient has an MTOC variant and kind of coming back to life and using that material. So what she did was take eggs from these animals and she vitrifies them or freezes them and then warms them back up. She then takes these eggs after recovery and she activates them to trick them into thinking that they've been fertilized to make parthenotes, and then develops them for 5 days to the blastocyst. And I can tell you, it really takes an embryologist to be able to do this type of work because it was a lot and she is so skilled, it's amazing. So here's what she saw. So she took a look at these blastocysts, and I hope you can appreciate that these homozygous blastocysts look a little bit different than the wild type. And what she noticed was that there were fewer blastocysts that reached this expanded blastocyst stage, something that an embryologist would score before transfer. And when she started looking at cell numbers, she saw a trend to having fewer cells in the homozygous blasts. And a trend again of having an altered ICM to trophectoderm ratio. So these aren't statistically significant, but when you're starting to think about human embryology, if this were to have an impact on a patient that has 4 blastocysts, this could start to become significant in their IVF success. So one of the things that we've been thinking about, and I get asked a lot in this project, is, well, how could CEP120 be affecting human oocyte spindle building? And the reason why we want to know this is that human oocytes build a spindle in a very different way than a mouse. And so we've been validating all of this in a mouse, and could there be some differences? So we know that in somatic cells, there is a pericentrin that localizes to centrosomes. And we know that in mouse oocytes, either at M1 or M2, pericentrin is also at the poles of these spindles. So mouse oocytes have really nice pericentriolar material, even though they lack centrioles. But Melina's lab showed a number of years ago that human oocytes don't have detectable pericentrin. And in fact, they think that the spindle is kind of being built inside out, coming off of the kinetochores and then clustering kind of at the ends of the spindles. And so our question is if human oocytes lack these MTOCs, do they even need CEP120? Probably the first experiment that maybe we should have done, but we like to do things backwards. So again, we turned to a tool that Melina's lab had built, and thank you, Melina, for developing all these amazing tools, where they can actually take mouse oocytes and humanize them into building a spindle. So what they do is they use TrimAway, and they inject in TRIM21 and an antibody that recognizes pericentrin. And that will acutely deplete pericentrin, and as you can see here in the picture, it actually gets rid of a number of the spindle pole and pericentriolar materials in a mouse oocyte, and they start building spindles in a very similar way to humans, and in fact you can see that they're quite short in these pictures. So that's what Marlena did. So she took our oocytes, either from wild type or our homozygotes, and she knocked down pericentrin and then assessed metaphase 2. And as a control, we inject in IgG. And we wanted to set up the system to look at metaphase 2 spindles. And so first, what Marlena did was take a look at M2 in her control cells, and you can see nice pericentriolar material looking at CEP192 and pericentrin. And in her depleted situation, she can't detect CEP192, and pericentrin is gone. What we found was that we actually had to take these oocytes and culture them for quite a bit longer than a mouse oocyte to reach metaphase 2, which typically takes about 14 to 16 hours. Here we had to carry out the experiment for 24 hours because this method of building a spindle, like in a human oocyte, is quite inefficient and it takes a lot longer. So then what Marlena wanted to know is there an impact on aneuploidy if you then do this experiment in her homozygous mutants? So we're going to do the in situ chromosome spread assay again. In the pericentrin intact oocytes, you see there's lots of pericentrin, and we see very low levels of aneuploidy in her wild type oocytes and a slight elevation in the homozygotes. When you get rid of pericentrin, you elevate aneuploidy in the wild type situation, and this is consistent with what has been reported in the literature. But importantly, we see a quite significant increase of aneuploidy in our homozygous mutants to about 60%. So in thinking about what CEP120 and this variant can be doing, ultimately we don't really know. We know that its role in somatic cells is cell cycle dependent. And what I mean by that is that when you knock out CEP120 in quiescent cells, this actually leads to increased microtubule nucleation. So it's suggesting it has some sort of inhibitory role. But most studies have looked at actively dividing cells and suggest that CEP120 actually has a positive role in microtubule nucleation. So I think that we still don't quite understand what could be going on, but we're wondering if it has something to do with affecting this dimerization domain. So to conclude, what I didn't show you and that we've published before is if you deplete acutely CEP120, we know that we elevate aneuploidy and we reduce microtubule nucleation. What I didn't show you is that we actually went on to make an oocyte-specific knockout of CEP120, and it was kind of disappointing to see that these were completely fine. And so there might be some compensation, possibly by polo-like kinase. From our previous study, when you overexpress this variant, we see elevated aneuploidy and reduced microtubule polymerization. And in our mutants, we see subfertility and again elevated aneuploidy and problems with microtubule nucleation. So where we're left with, and I think what we need to do with all of these MTOC variants is thinking about what this means for patients. So could mutation in CEP120 or these other MTOC genes, could these patients maybe not have as high of success rates if they're undergoing egg vitrification, cryopreservation, or even the embryo cryopreservation? And this really requires further study. And so our patients are heterozygous for this allele, and so we're really thinking, perhaps it's a heterodimer is affecting the ability to nucleate microtubules, or maybe it's acting in a dominant negative fashion and giving some sort of inhibitory signal to prevent polymerization. So we still don't really quite know. But to come back to all of these curves, you saw a lot of curves this morning. the way that we're thinking about this is that one's gene variants could really kind of determine what their aneuploidy curve might look like. So this is that average curve, but perhaps some of our patients actually reach these higher levels of aneuploidy a lot faster. Others might be kind of like shifted in time for earlier onset of aneuploidy. But where I think this CEP120 variant lies is that these patients might have a higher starting point of aneuploidy based on some of the analysis in our young mice, but then they reach this higher point a lot faster. It might be why we identified it in our patient cohort. So ultimately, our long-term goal is trying to understand reproductive genetic risk so that we hope to one day have some precision medicine that we can offer patients. So finally, I just want to thank the people that did the work. This is Marlena, super talented embryologist. I would want her making my babies. She's amazing. And to the NIH for their support. Thank you.
Thank you. We have some questions here.
Beautiful talk. This is not the part you focused on, but I'm really curious about the outliers who do better than expected, because they're still coming to the fertility clinic. So presumably they are either having a fertility issue or they're just there because of their age. And I'm wondering if you can see that, and if they have something else that's driving
so we
that?
don't have information on why these patients came. My guess is, because women can be referred to the clinic over the age of 35 after 6 months versus a year. That might be why they get there sooner. But aneuploidy-wise, they're quite okay. So maybe there's something with their partner that hasn't been diagnosed. But we've started looking at the gene variants in those patients to see if there is something that could be protective, although I liked how Eva said, well, maybe they have a lower burden of the harmful ones, and we haven't thought about it that way. But the protective variants are interesting because, at least in our cursory analysis, I don't think it's something that's actually directly on meiosis, and it might be something in the ovary instead. And so we're really trying to figure out how to best study those and test them, because it's, especially in a mouse system, it's hard to make a mouse do better than it already does. So we'll see.
Thank you.
Hi. I was curious about your oocyte-specific mutant not showing the aneuploidy phenotype, and I just wanted to ask, is that a straight knockout or is the point mutant?
It's a conditional knockout, so we used a GDF9 Cree on a floxed allele of CEP120. As far as we can tell, the protein is gone, but we've only looked by IF. But they look perfectly fine, like really perfectly fine. So, but in the literature, there's suggestion that like CEP120 and PLK1 are kind of acting in these parallel pathways. So I suspect that that's probably what's happening.
It's interesting that the point mutant might be a dominant negative or having a stronger effect.
Right, yeah, and I mean, we see that with some of our other genetic studies where when you completely get rid of a protein, others are like happily to step into line and do the job, whereas if you have some sort of mutation and the protein is still there, that compensation can't happen. So that's my best guess.
Hi, Karen. Maybe I missed it. Did you show where CEP120 localizes in human oocytes?
No.
And also, I was wondering whether, we saw yesterday how AlphaFold may be amazing to predict how a mutation can affect the structure. So I don't know if you looked into it more into understanding what the protein is doing.
Yeah, so great suggestion. So I haven't looked in human oocytes where it is. I can't remember if it's in Melina's paper, but it's in the human MTOC paper. And they see that CEP120 is at the poles. So it's definitely expressed, or like I shouldn't say poles, at the ends of the spindles in human oocytes. So it's definitely there in human. And when I was putting this together, because this has been just sort of this slow-moving project, and I thought, well, this is a good chance to like assemble something that we should get out there. I had the same thought. I was like, oh, we should totally do AlphaFold to look and see if this affects dimerization. So that is in the works, but I didn't put it together in time
But
to
do you
get
know,
that.
like, because I was wondering whether it relates to the RANGDP pathway, so do you know what potential interactive partners may be related to the pathway?
Yeah, no, it's great ideas. I do know that there's another mutation that other people have studied that's close by in that dimerization domain, and that does affect microtubule nucleation. So that's, you know, kind of where I am right now, but until we test that, I'm just hand waving. Yeah.
Really nice talk, Karen, really nice data. One point I'm always wondering about the PGTA data and aneuploïdy rates is how much are they affected by which embryos are selected for testing as well, and how consistent is this amongst clinicians, right? So I think probably they always underestimate aneuploïdy rates because probably only the best looking blastocysts are normally biopsied and selected, so probably overall the rates are lower than they would be across all embryos because you just disregard the worst ones. But then at the same time, like I don't know how much is known about this, but would embryologists always take the same ones, or would may some just take the very best looking ones, and then they would look, you know, they would end up higher in your graphs, and some are perhaps just saying, you know, I'll also take all the crappy ones, and then to which degree these data are also affected by how biopsies are being done and selected.
Yeah, so, and part of this is like this clinic. So this clinic really, we're one of the sort of leaders in being successful to culture embryos to the blastocysts. So these are only blastocysts biopsied. Certainly I think if they had biopsied any embryos that are rested, that would elevate probably everybody's aneuploïdy level. But this clinic is, the people who work there are quite good. I would imagine that they always sample sort of the same region for a control, but I don't know that for sure. And certainly this is a snapshot of you know, a patient's single cycle, and so depending on what the clinician may have done in a subsequent cycle, maybe their aneuploïdy rates have completely flip-flop, and that information we don't have either. But it gives us like a good place to start and like a career's worth of genes to work on, so it's good.
Thank you.
Okay, so we are right on time for the break. Thank you, everyone.